Thank you for your interest in my poster entitled Assessments for the In Vivo Tolerability and Efficacy of a JAK1-siRNA being developed for the highly targeted, infrequent, and efficacious treatment of inflammatory skin diseases using various application techniques, presented at the ACT 2025 conference in Phoenix, Arizona. My name is Shanté Jackson, Scientist, Study Director of Safety Assessment at Altasciences, and I will give you a brief overview of the work we have conducted. Atopic dermatitis and alopecia areata are just a couple of examples of inflammatory skin diseases, which are known to affect an estimated 20 to 25% of the population worldwide. Many current therapeutics in development for the treatment of these conditions fall into the class of small molecule Janus kinase or JAK inhibitors with a short half-life, which often necessitates daily treatments. JAK1 is a protein involved in the JAK-STAT signaling pathway, which regulates gene expression, cell growth, and immune responses. Downregulation or silencing of JAK1 can offer a therapeutic benefit for alleviation of cytokine storms linked to inflammatory skin diseases. Local inhibition of JAK1 in the skin provides the potential to achieve efficacy at the disease site while minimizing systemic drug exposure and associated toxicities. A small interfering ribonucleic acid, siRNA, that specifically targets JAK1, named ALD-102, is being developed as a highly targeted and efficacious clinical candidate for the infrequent treatment of inflammatory skin diseases. As part of that development, preclinical studies were designed with goals of evaluating the toxicity, tolerability, and efficacy of ALD-102. Gottingen mini pigs were administered both the clinical compounds, ALD-102, as well as the mini pig surrogate compound, ALD-105, to ensure pharmacologic activity in the test system for adequate assessments via intradermal injection and/or transepidermal delivery in the presence of star particles as a skin delivery enhancement technology. Star particles are submillimetric ceramic particles known to create temporary micropores in the stratum corneum, enhancing permeability and facilitating localized uptake of therapeutic molecules such as siRNA. For efficacy assessments, animals were administered a single intradermal injection of ALD-102 or ALD-105 or 5 millimolar ALD-102 containing 10% weight per weight star particles as a 3-day transepidermal loading regimen administered on days 1, 4, and 7. For toxicity and tolerability evaluation, animals were administered a supratherapeutic concentration of ALD-102 or ALD-105 at 43.04 mg per dose, 2 total doses once weekly. Administration of ALD-102 and ALD-105 was well tolerated across all evaluated dose levels and routes. Limited distal exposure or biodistribution of ALD-102 to off-target tissues was observed even at 43.04 mg per dose following repeat intradermal injections with minimal systemic exposure, supporting large safety margins for the clinical dose. A striking duration of local retention for ALD-102 and ALD-105 of 1 to 2 months was observed in mini pig skin following a single intradermal injection. Analysis of mini pig skin biopsies taken at various time points post-dose showed downregulation of target gene, JAK1 mRNA, and the downstream signaling pathway, CXCL9 and CXCL11. Out to at least 28 days following a single or intradermal injection. JAK1 mRNA inhibition was also sustained at week 4, 3 weeks following a transepidermal loading regimen of ALD-105 on treatment days 1, 4, and 7, when dose application was conducted in the presence of star particles. Overall, ALD-102 shows promise as a targeted, long-lasting treatment for inflammatory skin diseases based on intradermal delivery and efficacy results in mini pig skin 28 days post-injection. When combined with star particles, the combination overcomes skin barrier challenges and displays long-lasting efficacy sustained 3 weeks post a loading regimen. ALD-102 is currently in a phase 1b/2a trial via intradermal injection for alopecia areata patients.